Hello, my name is Abby Warren. I'm a recent graduate of Valdosta State University in the Department of Biology, and I worked with Dr. Yakov Waldman in the Department of Chemistry on Cytochrome C-based detection of superoxide. Superoxide is a free radical produced by the one-electron reduction of oxygen. It is considered a side product of the reduction of oxygen to water, but it's also been shown to be produced by immune cells as a chemical weapon to fight infection. We wanted to actually make a better probe to detect superoxide production in the cell, and we wanted to do that using cytochrome C. Cytochrome C is actually able to be reduced by superoxide, and when you look at cytochrome C's optical spectrum, you find that once it's reduced, its optical spectrum actually shifts, and you get a different reading. Now, optical spectrum actually is not as sensitive as we would like, so we decided to make a fluorescent probe instead of relying on the optical spectrum. So to do this, we had two main ways we wanted to go about it. The first was making cytochrome C mutants. We wanted to mutate the 4 lysines closest to the heme center, and we decided to mutate them to cysteine. We did this using overlap PCR, which you can see in the schematic of figure 3. We had one mutation for each of the 4 lysines we chose to mutate. We were going about this, and we got it down to having plasmids in the bacteria to produce the protein. We were having some difficulties with protein expression, however, it's recently been going better as this project has continued on. Our second type was modification of wild-type cytochrome C. We actually wanted to go about and dye it with the carboxyl X-rhodamine N-succinyl ester dye, and to do that we infused it slowly using a flow pump, and that was done over the course of several hours. Once we did that, we went ahead and purified it using chromatography, and we went through several purification steps in order to get as pure a protein as possible. We made sure that the dye and the protein ratio was the same before we combined all our fractions and did further purification. Once we had the final bit purified and as concentrated as we could get, we decided to compare it to wild-type cytochrome C and see how the spectrum versus the fluorescent readings were. We found that the rate of cytochrome C superoxide production by optical and the fluorescent methods were found to be about the same, and that validates our use of fluorescence because it shows that it's actually just as good, and we found the same rate while using fluorescence. So that means we can actually make this a valid probe, and then we can test sensitivity in actual oxygen producing systems. And that was going to be done at a later date.